Collagens are a family of proteins that are collectively the most abundant protein in the body and can be found throughout various connective tissues. Each member of the family is named with a Roman numeral, and if mutated or absent, it can lead to problems in the tissues where that particular collagen is found. Alport syndrome happens as a result of mutations in type 4 collagen, which is particularly important in the glomerulus of the kidney, the eye, and the cochlea, and that's why the symptoms of Alport syndrome are specific to those tissues. Type 4 collagen is a sheet-like structure found in all basement membranes and serves to support cells and form barriers. The three basement membrane layers are the lamina lucida, lamina densa, where type 4 collagen is, and lamina reticularis. Now, within the kidneys, there are glomeruli, which filter the blood and that, together with a tubule, form a nephron. These glomeruli happen to have basement membranes, called the glomerular basement membrane, or GBM. And that GBM, along with the fenestrated, meaning has pores, capillary endothelium, and the podocyte slit diaphragm, forms a selective filter, meaning that water and certain other plasma components can escape the capillary, forming the filtrate that will eventually become the urine. But red blood cells and most proteins stay in the glomerular capillary. In Alport syndrome, kidney functions normal through early childhood, but over time the missing or non-functional type 4 collagen causes the GBM to become thin and overly porous. And this allows red blood cells to pass right through the capillary to the urinary filtrate, leading to microscopic hematuria, which is where red blood cells are seen in the urine under a microscope. And this might eventually lead to gross hematuria, where the red blood cells can be seen with the naked eye. Over time, excessive amounts of protein start to get through the filter, resulting in proteinuria, or protein in the urine. And finally, this excessive protein loss and other factors cause the GBM to undergo sclerosis. As there are less and less healthy glomeruli, somebody with Alport syndrome might develop renal insufficiency, or even renal failure, which can lead to renovascular hypertension as well. Together, the hematuria, renal insufficiency, and hypertension contribute to the categorization of Alport syndrome as a glomerulonephritis. Another cause of glomerulonephritis, called Goodpasture syndrome, is caused by autoantibodies against the basement membrane in the glomeruli and the alveoli. So, although abnormality in the basement membranes from a different cause, the result is similar in these two syndromes. Another part of the body affected in Alport syndrome is the ear, which can lead to hearing loss, and usually this isn't present at birth, but starts in childhood or adolescence. And the precise mechanism behind this is unclear. One proposed mechanism involves the cochlea, or inner ear structure, which contains the organ of Corti, which has really small but highly specialized sets of hair cells that are attached to a basement membrane. Abnormal type 4 collagen might prevent those hair cells from generating normal nerve signals in response to sound vibrations. Because it's an inner ear problem, this is a type of sensory neural hearing loss, as opposed to middle ear and outer ear problems, which would lead to a conductive hearing loss. Some patients with Alport syndrome also have eye findings, which are also not typically present at birth. These include anterior lenticonus, where the middle part of the lens starts to push into the anterior chamber because the anterior lens capsule lacks the integrity to maintain the shape of the lens. Myopia or nearsightedness and lens opacities can result as well. Other eye changes in Alport syndrome include having white or yellow flecks around the macula, which is a central part of the retina, as well as having problems with the cornea, like recurrent erosions. Type 4 collagen is a heterotrimer, meaning that it's made up of 3 different polypeptide subunits, which are each composed of an alpha chain and that fit together in a triple helix. Each alpha chain has a very long repetitive amino acid sequence, with glycine followed by 2 variable amino acids, repeated over and over. Just remember that this is a repeat of amino acids, not a DNA triplet repeat. Now, glycine is the smallest amino acid, which allows it to fit tightly into the triple helix of collagen. If a mutation causes a larger amino acid to get put in place of glycine, then that regular helix can't be packed as tightly and might get degraded. Now, there are 6 similar alpha chains to choose from when building a collagen 4 trimer, which are encoded by 6 genes with the names that start with COL4 for collagen 4, and then an A since they code for the alpha chains, and a number 1 to 6 to designate which alpha chain. One version of type 4 collagen combines the alpha 3, alpha 4, and alpha 5 chains. Another combines two alpha 1s and an alpha 2. And a third version has two alpha 5s and an alpha 6. The alpha 1, alpha 2 collagen 4 is present in basement membranes at birth, but during infancy it gets replaced by the alpha 3, alpha 4, alpha 5 collagen 4, particularly in the glomerulus, the inner ear, and the eyes. When there's a mutation in the COL4A3, A4, or A5 genes, then the alpha 3, alpha 4, alpha 5 heterotrimer is affected, and this causes Alport syndrome. COL4A5 is on the X chromosome, so mutations in it can cause X-linked Alport syndrome, which presents early on and is the most common form of Alport syndrome. COL4A3 and A4 are on autosomes, meaning non-sex chromosomes, and mutations in these cause either autosomal recessive Alport syndrome, which is also early onset, or autosomal dominant Alport syndrome, which causes late onset disease. Also, mild mutations in COL4A3 and A4 can cause thin basement membrane nephropathy, where microscopic hematuria is the only symptom. For diagnosis, Alport syndrome is typically suspected when there are clinical signs like gross hematuria, or if there are vision or hearing problems, or microscopic hematuria with no apparent cause. A family history of Alport syndrome is also a good clue, but it also might be absent in cases of recessive Alport syndrome or with new spontaneous mutations. To confirm the diagnosis, a kidney or a skin biopsy is often analyzed by immunohistochemistry, meaning a labeled antibody is applied to a biopsy sample on a slide. Since misfolded collagen tends to get degraded, absent staining for collagen alpha chain suggests Alport syndrome. Skin is easier to biopsy, and although it contains the alpha 5, alpha 6 form of type 4 collagen, it doesn't have any alpha 3 or alpha 4 chains, so it can only be used to detect alpha 5 chain mutations. Alternatively, the biopsy can be looked at under an electron microscope, which early in the disease would show GBM thinning, while later in the disease would show a GBM with both thin and thick abnormal segments, and a lamina densa that appears split, with strands that look like a woven basket. Finally, some people might have gene testing for the COL4A genes. The treatment for Alport syndrome usually focuses on the symptoms. Proteinuria is treated with angiotensin-converting enzyme inhibitors and or angiotensin receptor blockers because there's evidence that this prevents progression to kidney failure. Anterior lenticonus can be treated with a replacement lens, and kidney failure might require dialysis or even a kidney transplant. Okay, as a quick recap, Alport syndrome is due to a mutation in gene COL4A, 3, 4, and 5, which encode for type 4 collagen alpha subunits 3, 4, and 5. And this causes kidney problems like hematuria and eventually glomerulonephritis, inner ear problems like sensorineural hearing loss, and eye problems like anterior lenticonus. Thanks for watching. You can help support us by donating on Patreon or subscribing to our channel or telling your friends about us on social media.